Hey there everyone, welcome back to another journey through the remarkable details of our natural world. Before we settle in for tonight's exploration, take a moment to hit that like button if you enjoy these deep investigations into the mysteries of biology and evolution, and subscribe if you want more content like this. Let me know in the comments where you're watching from and what time it is in your corner of the world. There's something fascinating about knowing we're all connected through these shared moments of curiosity, scattered across time zones and continents. Now, get comfortable, maybe dim the lights, put on some headphones, and let's ease into this investigation together. Tonight we're examining a question that has puzzled scientists, fascinated primatologists, and sparked countless myths since humans first began studying our closest living relatives. Why are chimpanzees so remarkably strong? What makes these animals, who share more than 98% of our DNA, capable of feats of strength that would leave even the most dedicated human bodybuilder trailing in their wake? The question seems simple enough on the surface. Chimpanzees are strong, everyone knows this. Popular culture has painted them as creatures of almost superhuman power, capable of ripping car doors off their hinges, snapping tree limbs like toothpicks, and overpowering grown men with terrifying ease. Stories circulate of chimpanzees in captivity demonstrating strength that seems to defy the laws of physics, pulling with forces that should be impossible for an animal that weighs on average between 40 and 70 kilograms. These aren't just urban legends either. Scientific experiments dating back to the 1920s have consistently shown that chimpanzees possess strength capabilities that exceed those of humans, sometimes dramatically so. But here's where the story gets interesting. For nearly a century, scientists couldn't quite explain why. The obvious answer seemed to be that chimpanzee muscles must simply be stronger, fibre for fibre, than human muscles. Perhaps their muscle tissue operated on fundamentally different principles, or maybe evolution had granted them some special biological advantage that allowed them to generate more force from the same amount of muscle mass. This seemed logical, intuitive even. After all, if chimpanzees are stronger, their muscles must be superior, right? Except they're not. At least, not in the way everyone assumed. When researchers finally managed to examine chimpanzee muscle fibers directly, measuring their contractile properties at the cellular level, they discovered something surprising. Individual chimpanzee muscle fibers, when isolated and tested in laboratory conditions, perform almost identically to human muscle fibers. They generate the same amount of force, contract at similar velocities, and operate according to the same basic physiological principles that govern muscle function across all mammals. The cells themselves, the fundamental building blocks of muscular strength, are essentially the same. This discovery didn't solve the puzzle. If anything, it deepened the mystery. If chimpanzee muscle fibers aren't inherently superior, then where does their remarkable strength come from? How can an animal generate 1.5 times the force of a human, sometimes more, using muscles that function identically at the cellular level? The answer, researchers have discovered, is far more fascinating than anyone imagined. It's not about having better muscles, it's about having different muscles, a different nervous system, a different skeletal framework, and a completely different body composition, all working together in ways that human bodies simply cannot replicate. Modern research has revealed that chimpanzee strength emerges from the intersection of 4 primary factors, each one contributing to their overall power in distinct but complementary ways. First, there's the composition of their muscle tissue itself, not at the level of individual fibers, but in terms of how those fibers are distributed and organized throughout their bodies. Second, there's the unique architecture of their nervous system, and the ways it controls muscle activation, operating on principles fundamentally different from human motor control. Third, there's the biomechanical framework provided by their skeleton, with bone structures and muscle attachment points optimized for generating force in ways human anatomy cannot match. And fourth, there's their overall body composition, the ratio of muscle to fat, the distribution of mass throughout their frame, and the metabolic strategies that support their physical capabilities. Understanding chimpanzee strength isn't just about satisfying curiosity, though that's certainly reason enough to investigate. It's a window into human evolution, revealing the trade-offs our ancestors made as they adapted to life on the African savanna. It illuminates the relationship between form and function, showing how subtle changes in biology can produce dramatic differences in capability. And it challenges our assumptions about strength itself, forcing us to reconsider what we mean when we talk about power, force, and physical capability. The story of chimpanzee strength is also the story of human specialization. We gave up raw power in exchange for something else, something that allowed us to become the dominant species on the planet despite being physically weaker than many of our relatives. Understanding what chimpanzees retained and what humans lost helps explain how we ended up so different, despite starting from such similar biological foundations. So tonight, we're going to examine each of these 4 factors in detail, exploring the science behind chimpanzee strength with the thoroughness it deserves. We'll look at muscle fibers under microscopes, trace neural pathways through spinal cords, measure moment arms and leverage, ratios, and calculate the metabolic costs of different body compositions. We'll meet the researchers who dedicated their careers to understanding these questions, review the experiments that finally provided answers, and piece together a comprehensive picture of what makes chimpanzees so remarkably, almost impossibly, strong. This is going to take some time. We're not rushing through this. Instead, we're settling in for a detailed exploration, moving methodically through each layer of complexity, building our understanding piece by piece until the full picture emerges. Because chimpanzee strength isn't a simple story with a single explanation. It's a convergence of multiple factors, each one fascinating in its own right, each one essential to understanding the complete picture. Before we begin, let's establish some baseline facts. Adult male chimpanzees in the wild typically weigh between 40 and 70 kilograms, roughly similar to human weights. Females are smaller, averaging between 27 and 50 kilograms. They stand about 150 centimeters tall, though they rarely adopt fully upright postures. Their bodies are covered in coarse black hair except for their faces, hands, and feet. They're omnivores, spending their days foraging for fruit, leaves, insects, and occasionally hunting smaller animals. They live in complex social groups, demonstrate remarkable intelligence, use tools and display emotions that humans find eerily familiar. And they're strong. Significantly, measurably, undeniably strong. Studies conducted over the past century have consistently shown that chimpanzees can generate forces approximately 1.5 times greater than humans of similar size when performing tasks like pulling or jumping. Some early experiments suggested even higher ratios, with claims that chimpanzees were 4 or 5 times stronger than humans, though more rigorous modern research has revised these estimates downward. The current scientific consensus places chimpanzee strength at roughly 1.35 to 1.5 times that of humans, a meaningful difference, though not as dramatic as popular accounts might suggest. But even this more modest multiplier is remarkable. It means that a 60-kilogram chimpanzee can match or exceed the pulling strength of a 90-kilogram human. It means that chimpanzees can perform athletic feats that would require extensive training for humans, and they do so with apparent ease. It means that in a direct physical confrontation, which thankfully almost never happens, a chimpanzee would have an overwhelming advantage, combining superior strength with fearlessness, aggression, and fighting tactics honed by millions of years of evolution. The question isn't whether chimpanzees are stronger. That's established fact, confirmed by countless experiments and observations. The question is why? What specific biological features produce this strength differential? How do chimpanzee bodies generate more force from similar-sized muscles? What evolutionary pressures shape their physical capabilities, and what does their strength tell us about our own evolutionary history? These are the questions we're here to answer. Over the next several hours of exploration, we're going to dismantle the myth of chimpanzee superstrength and replace it with something better, a detailed understanding of the actual mechanisms at work. We're going to look at muscle fibers through electron microscopes, measure electrical signals in motor neurons, calculate biomechanical leverage ratios, and examine the metabolic strategies that support different body compositions. We're going to truly understand why chimpanzees are so strong, and in the process we're going to learn something profound about what it means to be human, about the evolutionary trade-offs that made us who we are, and about the relationship between biology, behavior, and survival in the natural world. So let's begin. Let's start with the muscles themselves, with the fibers that generate force, with the cellular machinery that converts chemical energy into mechanical work. Let's examine what makes chimpanzee muscles different from human muscles, not at the level of individual cells, but in their overall composition and organization. let's understand the first major factor in chimpanzee strength, the unique architecture of their muscle tissue. Part 1. Muscle fiber composition. To understand why chimpanzees are so strong, we need to start at the cellular level, examining the basic units of muscular force, muscle fibers. These elongated cells are the fundamental machinery of movement, converting chemical energy stored in molecules of adenosine triphosphate into the mechanical force that moves our bodies through the world. Every time a muscle contracts, millions of these fibers are activating in coordinated waves, generating the force required to lift, pull, push, or hold against resistance. To understand why chimpanzees are so strong, we need to start at the cellular level, examining the basic units of muscular force, muscle fibers. These elongated cells are the fundamental machinery of movement, converting chemical energy stored in molecules of adenosine triphosphate into the mechanical force that moves our bodies through the world. Every time a muscle contracts, millions of these fibers are activating in coordinated waves, generating the force required to lift, pull, push, or hold against resistance. But not all muscle fibers are created equal. Decades of research into muscle physiology have revealed that skeletal muscle contains several distinct types of fibers, each optimized for different kinds of work. Understanding these fiber types, their distributions, and their properties is essential to understanding chimpanzee strength, because this is where the story begins. This is where chimpanzee muscles first diverge from human muscles, where the biological foundation for their superior strength is established. Let's start with the basics. Skeletal muscle fibers are classified according to the type of myosin, heavy chain protein they express. Myosin is the molecular motor that does the actual work of contraction, grabbing onto actin filaments and pulling them inward, shortening the muscle fiber and generating force. Different forms of myosin operate at different speeds and with different efficiency profiles, and the particular type of myosin a fiber expresses determines many of its functional characteristics. The primary distinction, the one that matters most for our discussion, is between what are commonly called slow-twitch and fast-twitch fibers. Technically, these are referred to as type 1 and type 2 fibers, or more precisely, fibers expressing myosin heavy chain 1 versus myosin heavy chain 2 isoforms. But the slow-twitch and fast-twitch terminology captures their essential difference quite well, so we'll use those terms here. Slow-twitch fibers, the type 1 fibers, are the endurance specialists of the muscle world. They contract relatively slowly, generating modest amounts of force, but they can maintain that contraction for extended periods without fatiguing. They're packed with mitochondria, the cellular power plants that produce energy, through aerobic metabolism, using oxygen to burn fat and carbohydrates efficiently. They're surrounded by dense networks of capillaries, ensuring a constant supply of oxygen-rich blood. They're rich in myoglobin, the protein that gives red meat its colour and helps store oxygen within the muscle tissue itself. These fibers are perfect for sustained repetitive activities. Walking, jogging, maintaining posture, performing repetitive work, anything that requires muscles to contract steadily over long periods, this is the domain of slow-twitch fibers. They don't generate explosive force, but they don't need to. They're optimized for endurance, for efficiency, for keeping going when fast-twitch fibers would have long since exhausted their energy supplies. Fast-twitch fibers, the type 2 fibers, are the opposite in almost every way. They contract rapidly, generating high forces in quick bursts, but they fatigue quickly. They rely primarily on anaerobic metabolism, breaking down glucose without oxygen in a process that produces energy quickly but inefficiently, generating lactic acid as a byproduct. They have fewer mitochondria, fewer capillaries, less myoglobin. They're paler in colour, giving white meat its characteristic appearance. They're built for power, not endurance, for explosive movements that demand maximum force generation in minimum time. Type 2 fibers actually come in several subtypes, distinguished by which specific myosin heavy chain 2 isoform they express. The main categories are type 2A, which are sometimes called fast oxidative fibers because they have some aerobic capacity, and type 2X or 2D, which are purely glycolytic and represent the fastest, most powerful but also most quickly fatigued fiber type. Different mammals and different muscles within the same mammal contain different proportions of these fiber types, reflecting the specific demands placed on those muscles by the animal's lifestyle and behaviour. Now here's where the story of chimpanzee strength begins to take shape. When researchers examined muscle tissue from chimpanzees and compared it to muscle tissue from humans, they found a striking difference in fiber type composition. Human muscles, across the trunk and limbs, contain approximately 70% slow-twitch fibers and only 30% fast-twitch fibers. Chimpanzees show the opposite pattern. Their muscles contain roughly 33% slow-twitch fibers and 67% fast-twitch fibers. The ratio is essentially reversed. This difference is profound. It means that when a chimpanzee activates a muscle, roughly 2/3 of the fibers generating force are fast-twitch fibers optimized for rapid, powerful contractions. In a human performing the same movement, only about 1/3 of the active fibers are fast-twitch, with the majority being slower, less powerful slow-twitch fibers. Right there, in that simple ratio, we find the first major explanation for chimpanzee strength. The discovery of this fiber type difference came relatively recently, published in a landmark 2017 study by Matthew O'Neill and colleagues from the University of Arizona College of Medicine. The research team obtained muscle tissue samples from chimpanzees, specifically from muscles in the pelvis and hind limbs, and used a technique called geol electrophoresis to identify the types of myosin heavy chain proteins present. They examined 35 different muscles, building a comprehensive picture of chimpanzee muscle composition. Then they compared their findings to existing data on human muscle composition, data gathered from similar anatomical locations using similar techniques. The results were unambiguous. Chimpanzees had significantly higher proportions of myosin heavy chain, 2 isoforms, the fast-twitch fibers, across all the muscles examined. The specific breakdown showed roughly equal distributions of type 1, type 2A, and type 2D fibers in chimpanzees, compared to the strong bias toward type 1 fibers in humans. This wasn't a subtle difference or a statistical quirk. This was a fundamental reorganization of muscle tissue, a wholesale shift in the cellular composition of the muscular system. To understand why this matters so much, we need to think about what happens when a muscle contracts. When your brain sends a signal to a muscle, telling it to generate force, that signal travels down motor neurons and triggers the activation of muscle fibers. In a maximal effort, where you're trying to generate as much force as possible, essentially all the fibers in the muscle activate simultaneously. In a chimpanzee, that means activating a muscle that's 2/3 fast-twitch fibers. In a human, it means activating a muscle that's 2/3 slow-twitch fibers. The fast-twitch fibers produce more force and produce it more quickly. They're designed for explosive power output, so when a chimpanzee and a human both maximally activate a muscle of the same size, the chimpanzee's muscle generates more force simply because more of its fibers are the high-force, high-speed variety. The individual fibers aren't stronger. Remember, fiber for fiber, chimpanzee and human muscle cells perform identically. But the composition of the muscle, the proportion of fast-twitch to slow-twitch fibers, creates a significant performance advantage for the chimpanzee. The researchers quantified this advantage using computer simulations. They built virtual muscle models incorporating the measured fiber type distributions, along with other known parameters like fiber length and maximum force generation capacity. Then they simulated various muscle performance tasks, like accelerating a load or performing cyclical contractions, and measured the maximum force and power output each model could produce. The chimpanzee muscle model consistently outperformed the human muscle model, generating approximately 1.35 times more dynamic force and power output. This ratio, 1.35, matches remarkably well with the observed performance differential between chimpanzees and humans in actual physical tests. When researchers compiled data from pulling and jumping experiments conducted over the past century, accounting for differences in methodology and subject size, they found that chimpanzees typically outperform humans by a factor of about 1.5. The muscle fiber composition difference accounts for most of this performance gap. The higher proportion of fast-twitch fibers in chimpanzee muscles explains, to a large degree, why chimpanzees are stronger. But the story doesn't end there. Fiber type distribution is just the beginning. The researchers also discovered that chimpanzee muscle fibers are longer, on average, than human muscle fibers. This might seem like a minor detail, but it has important implications for muscle performance. Longer fibers have a broader force-length relationship, meaning they can generate substantial force across a wider range of muscle lengths. This enhances the dynamic force, work and power capabilities of the muscle tendon unit, allowing chimpanzees to generate high forces throughout a greater range of motion. The longer fibers also mean that chimpanzee muscles can contract through larger distances, potentially increasing the work output per contraction. When combined with the higher proportion of fast-twitch fibers, which contract more rapidly, you get muscles that can generate high forces, contract quickly, and work effectively across a wide range of joint angles. These are muscles optimized for the demands of arboreal life, for climbing, swinging, and pulling the body weight through three-dimensional space, activities that require explosive power generation in constantly changing body positions. Now, you might be wondering why humans develop such a different muscle composition. If fast-twitch fibers are so powerful, why don't humans have more of them? The answer reveals something fundamental about human evolution and the trade-offs that shaped our species. Slow-twitch fibers aren't inferior to fast-twitch fibers, they're differently specialized. And the activities that came to dominate human evolution, the behaviors that distinguished our ancestors from other apes, favored the specialization that slow-twitch fibers provide. Consider what slow-twitch fibers are good at: sustained, repetitive activity, walking long distances, running for hours, maintaining posture, performing precise controlled movements. These are exactly the activities that became central to human survival as our ancestors moved out of the forests and onto the open savannas of Africa. Walking became our primary mode of transportation, and humans are exceptional walkers. We can cover enormous distances on foot, far more than any other primate, and we do so with remarkable energy efficiency. This efficiency comes largely from our muscle composition. Slow-twitch fibers are metabolically cheaper to operate than fast-twitch fibers. They use less energy per unit of force generated, particularly during prolonged activities. They rely on aerobic metabolism, which is far more efficient than the anaerobic glycolysis that powers fast-twitch fibers. This means that a human can walk all day, covering 20 or 30 kilometers, using less total energy than a chimpanzee would require to cover the same distance. Even though the chimpanzee could sprint much faster over short distances, the metabolic savings from having more slow-twitch fibers also freed up energy for other purposes. Human brains are extraordinarily expensive, metabolically speaking, consuming roughly 20% of the body's total energy budget, despite representing only about 2% of body mass. Supporting a brain of this size requires either consuming more calories or reducing energy expenditure elsewhere. Humans did both, but the shift toward more efficient slow-twitch dominant musculature was part of the equation. We traded some strength for endurance and metabolic efficiency, and that trade paid dividends that allowed for cognitive expansion. The shift towards slow-twitch fibers also enabled the development of fine motor control, though we'll explore that more in the next section. For now, the key point is that human muscle composition isn't a deficiency, it's an adaptation, a specialization for a different lifestyle than the one chimpanzees pursue. We lost some strength, yes, but we gained something that proved even more valuable for survival in our ecological niche. When the researchers compared chimpanzee and human fiber type distributions to those of other mammals, they found that humans are the outliers. Most terrestrial mammals have muscle compositions similar to chimpanzees, with substantial proportions of fast-twitch fibers. Lemurs, galagos, macaques, cats, dogs, horses all show fiber-type distributions that favor fast-twitch fibers, or at least maintain rough parity between fiber types. Only one other primate regularly shows more slow-twitch than fast-twitch fibers: the slow loris, a small lethargic primate that moves in slow motion through forest canopies. Humans are alone among active mobile mammals in having such an extreme bias towards slow-twitch fibers. This suggests that the shift in muscle composition was a specific human adaptation, something that happened along our evolutionary lineage after we split from the common ancestor we share with chimpanzees. Somewhere between 6 and 7 million years ago, our lineage began the transformation from an ape with chimpanzee-like muscles to a hominin with the slow-twitch dominant musculature that characterizes modern humans. The transition probably happened gradually, over millions of years, as our ancestors became increasingly committed to bipedalism and terrestrial travel. Each small increase in the proportion of slow-twitch fibers would have provided some advantage in terms of energy efficiency and endurance, advantages that compounded over evolutionary time. Natural selection favored individuals who could walk farther, forage more efficiently, and maintain activity levels throughout long days of travel and food gathering. Those individuals had slightly more slow-twitch fibers, slightly better endurance, slightly more efficient metabolisms. Their genes became more common in the population, and over countless generations the human muscle profile emerged. This evolutionary shift had costs, of course. We became weaker in absolute terms, less capable of explosive power output, less able to generate maximal force in single bursts. In an environment where physical confrontations with predators or competitors were common, this would be a serious disadvantage. But humans compensated for reduced strength through other means: better weapons, better tactics, better cooperation, better planning. We became weaker but smarter, and in the long run, the trade proved worthwhile. The muscle fiber composition story also connects to human athletic performance and the variations we see among individuals. Although all humans are slow-twitch dominant compared to chimpanzees, there's still variation within our species. Some people have slightly higher proportions of fast-twitch fibers, perhaps 35 or 40% instead of the 30% average. These individuals often excel at activities requiring explosive power: sprinting, jumping, throwing, weightlifting. Others have even more extreme slow-twitch dominance, perhaps 80 or 85% slow-twitch fibers, and these individuals tend to excel at endurance activities like marathon running or long-distance cycling. Training can shift fiber type distributions to some degree, though the changes are modest. Intense strength training can increase the proportion of fast-twitch fibers by perhaps 10 to 15%. Endurance training can do the opposite, slightly increasing slow-twitch fiber percentages, but these changes are small compared to the species-level differences we're discussing. No amount of training will give a human a chimpanzee-like muscle composition. The baseline is set by genetics, by evolutionary history, by the selective pressures that shaped our species over millions of years. It's worth noting that the fiber type difference between humans and chimpanzees isn't absolute. It's a population-level trend, a statistical difference in average compositions. Individual variation exists within both species. Some chimpanzees might have slightly fewer fast-twitch fibers than average, and some humans might have slightly more. But the overall pattern is clear and consistent. Chimpanzees are fast-twitch dominant, humans are slow-twitch dominant, and this difference contributes significantly to the strength differential between our species. The research into chimpanzee muscle composition also revealed something interesting about how muscle fibers are distributed within individual muscles. The researchers found that the fiber type ratios they measured were relatively consistent across different muscles, from the glutes to the quadriceps to the calf muscles. This suggests that the shift toward fast-twitch dominance affects the entire muscular system, not just specific muscle groups. It's a whole-body adaptation, a systemic reorganization of muscle tissue that affects virtually every muscle used in locomotion and manipulation. This consistency makes sense from an evolutionary perspective. Chimpanzees need explosive power throughout their bodies for climbing, not just in their arms or legs specifically. They need to generate rapid, forceful contractions in their shoulder muscles to pull themselves upward, in their hip and thigh muscles to push off from branches, in their hand and foot muscles to maintain grip and support body weight. A muscle composition optimized for power generation provides advantages across all these activities, so natural selection would favor individuals whose entire muscular system shifted toward fast-twitch dominance. The same logic applies to humans, but in reverse. We need endurance throughout our bodies for bipedal walking, not just in our legs. Our trunk muscles must maintain posture through hours of upright locomotion. Our arm muscles must carry tools and resources over long distances. Our neck muscles must support our heavy heads throughout extended periods of activity. Slow-twitch dominant musculature provides advantages across all these functions, so the shift towards slow-twitch fibers affected the entire human muscular system. Understanding muscle fiber composition also helps explain some specific differences in physical capabilities between humans and chimpanzees. For instance, chimpanzees are exceptional jumpers, capable of vertical leaps that would impress even trained human athletes. This jumping ability comes partly from their higher proportion of fast-twitch fibers, which can generate the explosive force needed to accelerate the body upward against gravity. Humans, with our slow-twitch dominant legs, simply cannot match this performance, even with training. Similarly, chimpanzees excel at pulling activities, whether pulling themselves up a tree or pulling against resistance in experimental settings. The pulling motion recruits large muscle groups in the back, shoulders, and arms, and when those muscles are 2/3 fast-twitch fibers, they can generate enormous forces in single bursts. Humans can develop significant pulling strength through training, building larger muscles with more total fibers, but we're working with a muscle composition that fundamentally limits our peak power output in these activities. On the other hand, humans excel at activities that favour slow-twitch fibers. We're better distance runners than chimpanzees by a wide margin. We can maintain relatively high speeds over extended periods, something no other primate can match. We're better at precise, controlled movements that require steady force output, activities like fine manipulation or careful tool use. We're better at maintaining posture and position through long periods, whether sitting, standing, or walking with loads. These advantages stem directly from our muscle fiber composition, from the same slow twitch dominance that limits our strength. Before we move on to the next major factor in chimpanzee strength, let's summarize what we've learned about muscle fiber composition. Chimpanzees have approximately 67% fast-twitch fibers and 33% slow-twitch fibers, while humans have approximately 30% fast-twitch fibers and 70% slow-twitch fibers. This reversed ratio means that chimpanzee muscles generate more force and power in explosive contractions, contributing significantly to their superior strength. The fiber type difference, combined with longer muscle fibers in chimpanzees, accounts for much of the observed 1.35 to 1.5 times strength differential between the species. This muscle composition difference is genetically determined, established during development, and relatively insensitive to environmental influences. It reflects an evolutionary divergence between the human and chimpanzee lineages, with humans shifting towards slow-twitch dominance as our ancestors adapted to bipedalism and terrestrial endurance activities, while chimpanzees retained the fast-twitch dominance common among primates and other terrestrial mammals. The shift involved trade-offs, with humans gaining endurance and metabolic efficiency at the cost of explosive strength, while chimpanzees retained strength at the cost of endurance capacity. This is the first piece of the puzzle, the foundation upon which chimpanzee strength is built. But it's not the complete story. Muscle composition explains much of the strength differential, but not all of it. To fully understand why chimpanzees are so strong, we need to examine the other factors at play. We need to look at how those muscles are controlled, how the nervous system activates muscle fibers and coordinates movement. We need to understand the second major factor in chimpanzee strength, the unique architecture of their motor control system. Part 2, motor control and nervous system muscles don't contract on their own. They require signals from the nervous system, electrical impulses transmitted along motor neurons that trigger the complex cascade of cellular events leading to fiber contraction. How these signals are generated, how they're distributed across muscle fibers, and how they're coordinated in time and space. All of this determines the actual force output that results from a given muscle composition. You can have the most powerful muscles in the world, but if your nervous system can't activate them effectively, that potential strength remains locked away, inaccessible. Muscles don't contract on their own. They require signals from the nervous system, electrical impulses transmitted along motor neurons that trigger the complex cascade of cellular events, leading to fiber contraction. How these signals are generated, how they're distributed across muscle fibers, and how they're coordinated in time and space. All of this determines the actual force output that results from a given muscle composition. You can have the most powerful muscles in the world, but if your nervous system can't activate them effectively, that potential strength remains locked away, inaccessible. This brings us to the second major factor in chimpanzee strength, their motor control architecture. The way a chimpanzee's nervous system activates and regulates muscle contraction differs fundamentally from the human pattern. These differences amplify the strength advantages provided by their muscle fiber composition, creating a neuromuscular system that generates force in ways human bodies simply cannot replicate. Understanding these neural differences requires examining motor neurons, spinal cord anatomy, cortical control, and the complex interplay between voluntary and reflexive movement patterns. Let's start with motor neurons, the nerve cells that directly connect to muscle fibers and trigger their contraction. Each motor neuron extends from the spinal cord to a muscle, where it branches and connects to multiple muscle fibers. This connection between a single motor neuron and all the muscle fibers it innervates is called a motor unit. When the motor neuron fires, sending an action potential down its axon, all the muscle fibers in its motor unit contract simultaneously. Motor units are the fundamental quantum of muscle activation, the smallest amount of force a muscle can generate in a controlled manner. The size of motor units varies tremendously across different muscles and different species. In muscles used for fine control, like the muscles that move the fingers or control eye movements, motor units are small, with each motor neuron connecting to only a handful of muscle fibers. This allows for precise gradation of force, with the nervous system recruiting more or fewer motor units to fine-tune the strength of contraction. In muscles used for powerful, less precise movements, like the large muscles of the legs and back, motor units are much larger, with each motor neuron connecting to hundreds or even thousands of muscle fibers. Here's where the first major difference between chimpanzees and humans emerges. Research by primatologist Anne McLarnon and evolutionary biologist Alan Walker revealed that chimpanzees have significantly less grey matter in their spinal cords than humans do, relative to body mass. Grey matter in the spinal cord contains the cell bodies of motor neurons, along with interneurons that process sensory information and coordinate movement patterns. More grey matter means more neurons, and in the case of motor control, more neurons means more motor units. Humans have more motor neurons than chimpanzees. This means that human motor units, on average, are smaller than chimpanzee motor units. Each human motor neuron connects to fewer muscle fibers, allowing for finer control over force output. Conversely, chimpanzee motor units are larger, with each motor neuron controlling more muscle fibers. When a chimpanzee motor neuron fires, it activates a larger fraction of the total muscle mass. The implications of this difference are profound. With smaller motor units, humans can engage just a few muscle fibers at a time, producing very small forces with great precision. We can thread a needle, write with a pen, perform microsurgery, because our motor units allow us to recruit tiny fractions of our muscle mass. We can gradually ramp up force output, recruiting more motor units as needed, smoothly transitioning from minimal effort to maximal exertion. Chimpanzees cannot do this. Their larger motor units mean that muscle activation is more of an all-or-nothing proposition. When a chimpanzee tries to generate force, each motor neuron that fires activates a large chunk of muscle tissue. There's less ability to fine-tune force output, less capacity for gentle, gradual recruitment. Instead, muscle activation in chimpanzees tends toward what researchers describe as an all-or-nothing pattern. Muscles are either relaxed or they're contracting forcefully. This pattern has an obvious implication for strength. When a chimpanzee needs to generate force, their motor units ensure that large fractions of their muscles activate quickly. There's less held in reserve, less fine gradation of effort, more commitment to full activation. In a maximal effort, a chimpanzee might activate a higher percentage of their total muscle mass than a human attempting the same movement. Their nervous system lacks the inhibitory control that allows humans to hold back, to engage only part of their strength while keeping reserves available. Alan Walker proposed that this difference in motor control might be even more significant than muscle composition in explaining chimpanzee strength. He suggested that humans have evolved greater cortical and spinal inhibition of muscle recruitment, neural mechanisms that prevent us from activating all our muscle fibers simultaneously. This inhibition serves multiple purposes. It prevents tissue damage from excessive force generation, it allows for fine motor control, and it enables the smooth, coordinated movements that characterize human manipulation and tool use. But this inhibition also limits our strength. We're neurally prevented from recruiting all our muscle fibers in a single burst, even when we're consciously trying to generate maximum force. There are countless anecdotes of people performing superhuman feats of strength in emergency situations, lifting cars to free trapped victims or breaking through barriers they couldn't normally budge. These stories, if true, probably reflect temporary suspension of the neural inhibition that normally limits our strength. Under extreme stress, when adrenaline floods the system and survival instincts override normal controls, some of the neural limiters might release, allowing brief access to strength reserves normally kept locked away. Chimpanzees, according to Walker's hypothesis, lack this same degree of neural inhibition. Their motor control system doesn't prevent them from recruiting large fractions of their musculature. They can access a higher percentage of their potential strength during normal, voluntary movements. This doesn't mean they're always operating at maximum capacity. Obviously, chimpanzees modulate their force output according to the demands of the task at hand, but their range of modulation is different from ours, skewed toward higher force outputs, with less fine control at the low end. There's an evolutionary logic to this difference. Fine motor control is essential for human behaviours: tool manufacture, precise manipulation, throwing, writing, all the activities that define human culture and cognition. We need to be able to engage small fractions of our muscles with great precision. For chimpanzees, fine motor control is less important. They use tools, yes, but not with the precision or complexity that characterises human tool use. They don't throw with accuracy. They don't need to write or perform microsurgery. Their lifestyle emphasises explosive power, climbing, fighting, moving rapidly through three-dimensional spaces. For these activities, all-or-nothing motor control is actually advantageous, allowing rapid mobilization of strength without the delays introduced by gradual recruitment. However, it's important to note that the neural inhibition hypothesis remains somewhat controversial. While Walker's proposal is elegant and fits well with what we know about chimpanzee behavior and human capabilities, direct experimental evidence is limited. The O'Neill study that revealed the muscle fiber composition differences explicitly tested whether neural factors might contribute to the strength differential. Their conclusion was that humans are capable of complete or near-complete voluntary activation of their musculature when performing maximal effort tasks, suggesting that neural inhibition might not limit human strength as much as Walker proposed. The debate continues, with researchers arguing about the relative contributions of muscle composition versus neural control to the chimpanzee-human strength differential. The current consensus seems to be that muscle composition, specifically the fiber type distribution we discussed in Part 1, accounts for most of the difference, but that neural factors might contribute as well, perhaps adding another 10 or 15% to the chimpanzee advantage. More research is needed to fully resolve this question, particularly studies that directly measure motor unit recruitment patterns in both species during maximal effort tasks. Regardless of the exact magnitude of the neural contribution, it's clear that motor control patterns differ between humans and chimpanzees in ways that affect strength output. These differences extend beyond motor unit size and recruitment patterns to encompass the entire architecture of the motor control system, from cortical planning centers to spinal circuits to the neuromuscular junctions where motor neurons connect to muscle fibers. Let's examine some specific aspects of this motor control architecture. First, consider the role of muscle synergies. Muscle synergies are coordinated patterns of muscle activation that the nervous system uses as building blocks for complex movements. Instead of individually controlling each muscle, the brain activates pre-programmed synergies that coordinate multiple muscles simultaneously. This reduces the computational burden of motor control, allowing the nervous system to generate complex movement patterns without having to micromanage every detail. Research on muscle synergies in primates has revealed that chimpanzees and humans both use synergistic control, but the specific synergies and how they're combined differ between species. Studies examining electromyographic activity during locomotion in chimpanzees show that they rely on relatively few, robust synergies that coordinate large groups of muscles in stereotyped patterns. These synergies are shared across different locomotor modes: quadrupedal walking, bipedal walking, and vertical climbing, all utilize similar underlying synergy patterns with modifications for the specific demands of each behavior. This synergistic organization is consistent with the all-or-nothing motor control we've been discussing. Chimpanzee movement patterns are robust and powerful, coordinating large muscle groups in forceful, stereotyped ways. Human synergies, by contrast, appear to be more numerous and more flexible, allowing for greater variety in movement patterns and finer control over specific muscle groups. We've traded some of the robustness and power of chimpanzee movement for greater flexibility and precision. The neural circuits that generate these synergies reside partly in the spinal cord itself, in networks of interneurons that coordinate motor neuron activity. These spinal circuits can generate complex movement patterns without direct input from the brain, though they're normally modulated and controlled by descending signals from cortical and subcortical motor centers. The architecture of these spinal circuits differs between chimpanzees and humans, reflecting our different motor demands and movement patterns. Research on primate spinal circuits has shown that premotor interneurons in the cervical spinal cord The neurons that connect to motor neurons controlling arm and hand muscles are organized in clusters that correspond to muscle synergies. These clusters represent the neural substrate of synergistic control, the hardware that implements coordinated muscle activation. The organization of these clusters, the connections between them, and their modulation by descending pathways all contribute to species-specific motor control capabilities. In primates, including humans and presumably chimpanzees, these spinal circuits are overlaid with direct cortical control via the corticomotor-neuronal pathway. This pathway provides direct connections from motor cortex neurons to spinal motor neurons, bypassing the interneuron circuits and allowing for voluntary control over individual muscles. The corticomotor neuronal pathway is particularly well developed in humans, contributing to our exceptional fine motor control, especially in the hands and fingers. But this direct cortical control comes with a cost. The more the nervous system relies on top-down voluntary control, the less it can depend on automatic reflexive circuits. Chimpanzees, with their more robust spinal circuits and less extensive corticomotor neuronal pathways, can rely more heavily on automatic movement patterns that coordinate large muscle groups in powerful synergies without requiring conscious attention or fine-tuning. This makes their movements more forceful and more automatic, but less flexible and less precise. The different motor control strategies also affect how force is modulated during movement. Humans excel at smoothly varying force output, gradually increasing or decreasing the strength of muscle contractions in response to changing demands. This smooth modulation requires fine control over motor unit recruitment, the ability to add or subtract small numbers of motor units to tune force output precisely. It's essential for activities like writing, where we need to maintain constant pressure on a pen while moving the hand in complex patterns, or for manipulating fragile objects without breaking them. Chimpanzees are less adept at this kind of smooth force modulation. Their motor unit structure and control architecture favour more discrete force outputs. Either the muscle is relaxed, or it's contracting forcefully. There are intermediate levels, obviously, but the granularity of control is coarser than in humans. This makes chimpanzees less skilled at activities requiring delicate force control, but more effective at activities requiring rapid mobilization of strength. Another aspect of motor control that differs between humans and chimpanzees involves proprioception and force feedback. Proprioception is the sense of body position and movement, mediated by specialized receptors in muscles, tendons, and joints that detect stretch, tension, and position. This sensory information feeds back to the nervous system, providing the data needed to adjust motor commands in real time, maintaining balance, coordinating movements, and matching force output to task demands. Humans have highly developed proprioceptive systems, particularly in the hands and arms, where dense networks of sensory receptors provide detailed information about position, force, and texture. This rich proprioceptive feedback enables the fine motor control that characterizes human manipulation. We can adjust grip force based on the texture of an object, modify tool use patterns based on subtle changes in resistance, perform complex sequences of movements guided by tactile and proprioceptive cues. Chimpanzees also have sophisticated proprioceptive systems, but the use of proprioceptive information in motor control might differ from the human pattern. With their larger motor units and more automated movement patterns, chimpanzees might rely less on continuous proprioceptive feedback for fine-tuning muscle activation. Their motor control might be more feed-forward, based on pre-programmed movement patterns that execute largely without online modification, rather than the closed-loop feedback control that characterizes much of human movement. This difference would make sense given their different behavioral demands. Human activities like tool manufacture require constant adjustment based on sensory feedback, continuous fine-tuning of movements in response to how the tool and material respond. Chimpanzee activities like climbing require powerful, rapid movements that execute too quickly for much online adjustment. You grab a branch, pull hard, trust that your motor programs will generate the right forces. There's less time for feedback-based modulation, more reliance on robust pre-programmed patterns. The vestibular system, which detects head movement and orientation and contributes to balance and postural control, also plays a role in motor control differences between humans and chimpanzees. Humans have vestibular systems highly tuned for bipedal balance, providing continuous information about head position and movement that helps maintain upright posture. Chimpanzees have vestibular systems optimized for their more varied locomotor repertoire, providing information useful for climbing and brachiating as well as terrestrial movement. The integration of vestibular information with motor commands affects how strength is deployed during movement. Maintaining balance while generating force requires coordination between postural muscles and the muscles directly performing the task. Humans, with our precarious bipedal stance, need to carefully manage this coordination, ensuring that forceful movements don't destabilize posture. Chimpanzees, with their lower center of gravity and more stable quadrupedal posture, can generate force more freely without as much concern for postural stability. This brings us to an interesting aspect of chimpanzee strength, their low center of gravity. While not strictly a neural factor, the center of gravity affects how the nervous system can deploy muscular force. Chimpanzees have relatively short legs and long arms, with much of their body mass carried forward and low to the ground. This configuration provides inherent stability, making it harder to knock a chimpanzee off balance. When combined with their powerful muscles, this stability allows them to generate enormous forces without risking a fall. Humans, by contrast, have a high center of gravity perched atop long legs. We're inherently unstable, constantly making small postural adjustments to maintain balance. This instability constrains how we can use our strength, generate too much force in the wrong direction, and we topple over. Pull too hard on something above us, and we lose our balance. Our nervous system has to constantly account for these stability constraints, limiting force output to levels that don't compromise posture. Chimpanzees don't face these same constraints. Their low, stable posture allows them to pull, push, and generate force in almost any direction without losing balance. Their nervous system can commit more fully to force generation because it doesn't need to reserve as much capacity for postural control. This is part of why chimpanzees excel at pulling activities. They can plant themselves, generate maximum force with their upper body, and their stable base prevents them from being pulled off balance. The motor control story also intersects with muscle fatigue and recovery. We've already discussed how fast twitch fibers fatigue more quickly than slow twitch fibers. The neural control of muscle activation affects how this fatigue manifests, and how the nervous system compensates for declining force output as fatigue sets in. Humans, with their fine gradation of motor unit recruitment, can respond to fatigue by recruiting fresh motor units to replace fatigued ones, maintaining relatively steady force output even as individual muscle fibers tire. Chimpanzees, with their larger motor units and more all-or-nothing recruitment pattern, have less flexibility in how they compensate for fatigue. When motor units fatigue, there are fewer reserves to call upon, and force output drops more precipitously. This is consistent with observations of chimpanzee behavior. They're capable of brief explosive exertions but tire quickly if the effort continues. Their motor control strategy is optimized for short bursts of maximum force, not for sustained efforts where fatigue management becomes important. The different motor control strategies also have implications for motor learning and skill acquisition. Humans are exceptional at learning new motor skills, refining movement patterns through practice, gradually improving precision and efficiency. This learning involves both cortical changes, as motor cortex representations are refined, and spinal changes, as muscle synergies are modified and new synergies are established. Our flexible motor control architecture, with its extensive corticomotor neuronal pathways and capacity for fine gradation of force, enables this learning. Chimpanzees can also learn motor skills, of course, and they demonstrate impressive flexibility in tool use and manipulation. But the nature of their motor learning might differ from ours, focusing more on selecting among existing robust synergies rather than fine-tuning force output within synergies. Their motor repertoire might be more constrained, with less potential for developing entirely new movement patterns, but more reliability in executing the patterns they do possess. Recent research has begun to explore the genetic and developmental factors that shape motor control architecture. Genes, regulating motor neuron development, synapse formation, and spinal circuit assembly all contribute to establishing the species-specific motor control capabilities. Differences in the expression or regulation of these genes between humans and chimpanzees might explain some of the neural differences we've been discussing. Understanding these genetic factors could reveal when in evolutionary time the human and chimpanzee motor control systems diverged, and what selective pressures drove their different developmental programs. One particularly interesting gene family involves the homobox genes, which regulate positional identity during development, essentially telling cells where they're in the body and what structures they should form. Variations in homobox gene expression patterns can alter spinal cord organization, changing the number and connectivity of motor neurons. It's possible that human-specific changes in homobox gene regulation contributed to our altered motor control architecture, increasing motor neuron numbers and enabling finer control. The development of the corticomotor neuronal pathway in humans also involved evolutionary changes, likely related to brain development and expansion of the motor cortex. The human motor cortex is notably enlarged compared to chimpanzees, both in absolute size and relative to body size. This expansion provided the neural substrate for more elaborate motor control, more extensive cortical representation of individual muscles, more capacity for voluntary modulation of movement patterns. But brain expansion isn't free, metabolically speaking. The human brain consumes enormous amounts of energy, roughly 300 to 400 calories per day just to maintain basic function, more when engaged in intensive cognitive activities. Supporting this metabolic demand required evolutionary changes in diet, digestion, and energy allocation. Some of the same factors that enabled our altered muscle composition, the shift toward more efficient slow-twitch dominant muscles, also supported our brain expansion by freeing up energy that could be devoted to neural tissue. The motor control story also connects to another uniquely human capability: throwing. Humans are exceptional throwers, capable of launching projectiles with high velocity and remarkable accuracy. No other primate comes close to matching human throwing performance. This capability depends on a specific combination of anatomical and neural features, shoulder and arm proportions that allow for large ranges of motion, muscle activation patterns that coordinate the entire body in a kinetic chain, and motor control precision that enables accurate targeting. Chimpanzees can throw, but they do so poorly. They can't generate the same velocities, and their accuracy is terrible. Part of this limitation is anatomical. Their shoulder structure doesn't permit the same range of motion that humans achieve. But part is neural. Effective throwing requires precise timing of muscle activation sequences, coordinating muscles from the legs through the torso to the shoulder and arm in a cascade that transfers energy efficiently to the projectile. This kind of coordination demands fine motor control, the ability to time activations with millisecond precision, something that's difficult with the larger motor units and more automated control that characterizes chimpanzee motor systems. The inability to throw effectively limited chimpanzee weapon use and likely affected their evolutionary trajectory. Without effective throwing, hunting at a distance is impossible, and defense against predators is more difficult. Humans, with our superior throwing capability, could hunt more effectively, defend territories more successfully, and eventually develop elaborate projectile weapon technologies that amplified our capabilities even further. Our motor control system, with its emphasis on precision over pure force, enabled these technological and behavioral developments. The study of motor control in chimpanzees and humans also opens fascinating questions about our extinct relatives. What motor control architectures did Neanderthals have? What about Homo erectus or Australopithecus? Did the id shift toward finer motor control and more motor neurons happen gradually over millions of years, or was there a rapid transition at some point? Unfortunately, nervous tissue doesn't fossilize, so we can't examine spinal cord anatomy or brain structure directly in extinct species. But we might be able to infer some information from the size of the spinal canal, which houses the spinal cord, or from endocasts that reveal brain size and surface features. For instance, if we could measure the diameter of the spinal canal in fossil hominins, we might estimate how much grey matter they had and thus how many motor neurons. This could tell us whether they had human-like fine motor control or more chimpanzee-like all-or-nothing control. Similarly, examining the size and shape of the motor cortex in fossil skulls could reveal when the expansion of cortical motor control occurred in human evolution. These anatomical clues, combined with archaeological evidence of tool use and manufacture, could paint a picture of how motor control capabilities evolved in our lineage. The archaeological record shows a gradual increase in tool complexity over the past 3 million years, from simple stone flakes to elaborate hafted spear points, finely made needles, and eventually pottery and metalworking. Each advance in tool technology probably required corresponding advances in motor control precision. Early stone tools could be made with relatively coarse motor control, using powerful strikes to flake rocks. But the elaborate pressure flaking seen in later stone tools requires extremely fine force control, the ability to apply precisely graded pressure at specific angles. The appearance of such tools in the archaeological record might mark the point where human motor control capabilities had evolved sufficiently to enable these techniques. Similarly, the appearance of throwing spears in the archaeological record around 400,000 years ago might mark the point where human throwing capability, driven by improvements in motor control precision, had reached levels sufficient for effective hunting. The earliest spears are simple wooden points, but later examples show increasingly sophisticated hafting and balancing, suggesting refinement of throwing technique enabled by increasingly precise motor control. The evolution of human handwriting provides another window into motor control capabilities. The earliest writing systems appeared only about 5,000 years ago, a blink of an eye in evolutionary time, but they required motor control precision that probably evolved much earlier. The ability to make controlled, repetitive, fine movements with the fingers, to maintain steady pressure while moving the hand in complex patterns, to coordinate vision with hand movement for accurate placement. All these capabilities predate writing by hundreds of thousands of years, evolved for other purposes but subsequently co-opted for this cultural innovation. The motor control story reminds us that strength isn't just about muscles, it's about the entire neuromuscular system, the integrated function of muscle tissue, motor neurons, spinal circuits and brain centers, all working together to generate coordinated movement. Chimpanzees have a motor control system optimized for powerful, explosive movements, rapid mobilization of large muscle groups, automatic execution of robust movement patterns. Humans have a motor control system optimized for precision, flexibility, fine gradation of force, and voluntary control over individual muscles. Neither system is superior in absolute terms. Each is perfectly suited to the behavioral demands and ecological niche of its species. Chimpanzees need power for climbing, fighting, and rapid movement through complex arboreal environments. Their motor control system delivers that power efficiently, with minimal neural overhead, allowing for fast reflexive responses and maximal force output. Humans need precision for tool manufacture, throwing, and the countless delicate manipulations that characterize our technology-dependent lifestyle. Our motor control system delivers that precision, even though it means sacrificing some peak strength and automated responses. The trade-offs between precision and power, between voluntary control and automatic responses, between fine gradation and all-or-nothing activation, these are fundamental constraints that shape motor control evolution across all species. Every animal must balance these competing demands, optimizing for the specific challenges it faces. Chimpanzees and humans represent two different solutions to similar starting conditions, two different paths taken from a common ancestor, two different sets of trade-offs that led to dramatically different capabilities. Understanding these trade-offs helps us appreciate both species more fully. Chimpanzees aren't primitive versions of humans stuck at an earlier stage of evolution. They're fully modern species, as evolved as we are, optimized for their particular ecological niche through millions of years of natural selection. Their motor control system is sophisticated and effective, perfectly suited to their needs. It's different from ours, not inferior to ours. Similarly, humans aren't superior to chimpanzees simply because we have more neurons or finer control. We're differently specialized, adapted for different challenges, successful in different ways. Our motor control system enables remarkable precision but limits our strength. Their motor control system enables remarkable strength but limits their precision. Neither is the right solution. Both are successful solutions to different problems. Before we move to the third major factor in chimpanzee strength, let's summarize what we've learned about motor control. Chimpanzees have fewer motor neurons than humans, resulting in larger motor units that activate more muscle fibers per neural firing. They might have less neural inhibition-limiting muscle recruitment, allowing them to access higher percentages of their potential strength. They rely more heavily on robust spinal circuits and automated movement patterns, less on fine cortical control. They have a lower center of gravity that provides stability during force generation. They lack the extensive corticomotor neuronal pathways that give humans fine voluntary control, but also limit our peak force output. All these factors combine to maximize the force output from their muscle composition, adding to the strength advantages we discussed in part 1. This is the second piece of the puzzle, the neural component that determines how muscles are actually used. But we're still not done. Muscles and nerves don't operate in isolation. They're attached to bones, working through joints, leveraging mechanical advantage to convert muscular force into movement of body segments through space. To fully understand chimpanzee strength, we need to examine these mechanical factors, the skeletal architecture and biomechanical leverage that determine how efficiently muscular force translates into pulling, climbing, and manipulating the physical world. Let's examine the third major factor in chimpanzee strength, their skeletal biomechanics. Part 3. Skeletal architecture and biomechanics. Muscles generate force, but bones determine how that force moves the body. The skeletal system provides the rigid framework against which muscles can pull, the levers that amplify or reduce muscular forces, and the joints that determine which movements are possible and which are constrained. Understanding strength requires understanding mechanics, the physical principles that govern how forces propagate through rigid bodies and produce movement. And when we examine chimpanzee skeletal architecture through the lens of biomechanics, we discover the third major factor contributing to their remarkable strength. Let's start with the fundamental concept in biomechanics, leverage. When a muscle contracts, it pulls on a bone, causing that bone to rotate around a joint. The amount of rotational force, or torque, produced depends on two things: the amount of force the muscle generates and the leverage the muscle has around the joint. Leverage is determined by the moment arm, the perpendicular distance from the joint center to the line of action of the muscle force. Longer moment arms mean more leverage, allowing a given muscle force to produce more torque and thus more rotational power. This is where anatomy becomes destiny. The specific locations where muscles attach to bones, the lengths of bones, the positions of joints, all these factors determine moment arms and thus leverage. Small changes in skeletal architecture can have large effects on mechanical advantage, amplifying or diminishing the force output that results from a given amount of muscular contraction. Evolution has shaped skeletal architecture in both humans and chimpanzees to optimize the mechanical properties of the musculoskeletal system for their different behavioral demands. Chimpanzees have longer moment arms for many of their upper limb muscles compared to humans. This has been documented in detailed anatomical studies that measured muscle attachment sites, bone lengths, and joint positions in both species. For instance, studies by Susan Thorpe and colleagues measured the dimensions and moment arms of both forelimb and hind limb muscles in common chimpanzees, creating comprehensive datasets that allow for detailed biomechanical comparisons with human anatomy. The longer moment arms in chimpanzee forelimbs are particularly notable. Muscles like the biceps, which flex the elbow, and the latissimus dorsi, which pulls the arm backward and downward, have attachment points that provide greater leverage than the corresponding human attachments. This means that when a chimpanzee and a human generate equal amounts of muscular force, the chimpanzee's skeletal architecture converts that force into more torque at the joints, producing more rotational power and thus more pulling or climbing capability. Let's walk through a specific example to make this concrete. Consider the biceps muscle, which flexes the elbow. In humans, the biceps tendon attaches to the radius bone a few centimeters below the elbow joint. In chimpanzees, this attachment point is located slightly further from the joint, providing a longer moment arm. Computer models suggest this difference in moment arm is perhaps 10 to 20% larger in chimpanzees. This might not sound like much, but remember that torque equals force times moment arm. A 20% longer moment arm means 20% more torque for the same muscular force, a significant mechanical advantage. When we multiply this effect across all the muscles involved in pulling activities, the muscles of the shoulder, back, arms, and hands, the cumulative advantage becomes substantial. Chimpanzees aren't just generating more force with their muscles due to their fiber composition and motor control, they're also converting that force more efficiently into mechanical work due to their skeletal architecture. The result is pulling capabilities that far exceed what their muscle mass alone would predict. The skeletal advantages aren't limited to moment arms. Bone lengths also matter, affecting the ranges of motion and mechanical properties of limbs. Chimpanzees have relatively long arms compared to their body size, with arm lengths that exceed leg lengths. This is the opposite of the human pattern, where legs are longer than arms, Long arms provide several advantages for arboreal activities like climbing and reaching, but they also affect leverage in complex ways. Long limbs generally reduce mechanical advantage at the joints, because muscles have to work through larger ranges of motion to produce the same amount of limb displacement. However, long limbs also increase the potential work output per contraction, since the muscle force acts over a longer distance. The net effect on strength depends on the specific task and how forces are applied. For pulling activities, where the goal is to generate high forces rather than rapid movements, the chimpanzee arm proportions appear to provide advantages, combining long moment arms for key muscles with limb lengths appropriate for generating sustained pulling forces. The shoulder girdle in chimpanzees also differs significantly from the human pattern in ways that affect strength. Chimpanzees have more superiorly oriented scapula, shoulder blades that sit higher on the back and are oriented more toward the head than in humans. This orientation changes the lines of action for shoulder muscles, particularly the deltoids and rotator cuff, optimizing these muscles for overhead activities like climbing and brachiation. When a chimpanzee raises its arms overhead to climb, its shoulder muscles are operating at mechanical advantages that humans simply cannot achieve. The superior orientation of the scapula means that muscles are pulling at angles that maximize torque production in the overhead position. Humans, with our laterally oriented scapulae, have shoulder muscles optimized for different activities, particularly for manipulating objects in front of the body and below shoulder height. We can raise our arms overhead, obviously, but we do so less efficiently, with shoulder muscles working at less favorable angles. The acromion process, a bony projection on the scapula, also differs between humans and chimpanzees. In humans, the acromion projects laterally over the glenohumeral joint, the ball and socket joint that connects the humerus to the scapula. This lateral projection increases the moment arms for the deltoid muscles, providing mechanical advantage for raising the arm to the side, a movement important for throwing and tool use. In chimpanzees, the acromion is less laterally projecting, trading some mechanical advantage for sidearm movements in exchange for better overhead capability. These shoulder differences reflect the different demands placed on the upper limbs in the two species. Chimpanzees use their arms primarily for climbing, swinging, and manipulating objects above their heads. Their shoulder architecture is optimized for these overhead activities. Humans use our arms primarily for manipulating objects in front of our bodies, for throwing, and for carrying. Our shoulder architecture is optimized for these activities, even though it means less efficiency for overhead work. The pelvis and lower limbs also show significant architectural differences, though these affect strength less directly than the upper limb differences. Chimpanzees have broader, shallower pelvises than humans, with differently oriented iliac blades and ischia. These pelvic differences reflect adaptations for different locomotor modes. Chimpanzees maintain flexibility for climbing and quadrupedal walking, while humans have pelvises optimized for bipedalism. The muscle attachment sites on the chimpanzee pelvis provide different leverage for hip and thigh muscles, affecting their capabilities in climbing and jumping. The spine is another site of significant architectural difference. Humans have an S-shaped spinal curvature, with lordotic curves in the lumbar and cervical regions and kyphotic curves in the thoracic and sacral regions. This S-shape is an adaptation for bipedalism, helping to position the head over the pelvis and absorb shock during walking. Chimpanzees have a simpler spinal curvature, essentially maintaining the primary kyphotic curve present in fetal development. They also have one fewer lumbar vertebra than humans, resulting in a stiffer lower back. The stiffer spine in chimpanzees affects how forces are transmitted through the body. When climbing, a rigid trunk helps transfer forces from the limbs through the body core, providing stability during overhead pulling. When hanging, a stiff spine helps maintain body position without requiring excessive muscular effort. Humans, with our more flexible spines, have greater range of motion in the trunk, but potentially less efficient force transmission during climbing and pulling activities. The hands and feet of chimpanzees also show architectural differences relevant to strength. Chimpanzee hands have longer, more curved fingers than human hands, with muscle and tendon arrangements optimized for powerful gripping. Their thumbs are shorter and have different ranges of motion, providing less dexterity than human thumbs, but more strength in basic grip functions. Studies of thumb muscle cross-sections and moment arms have shown that while human thumbs have advantages for precision grips, chimpanzee thumbs generate higher forces in power grips. The foot architecture in chimpanzees includes an opposable big toe, essentially a thumb-like structure that allows them to grip with their feet much as they grip with their hands. This provides enormous advantages for climbing, allowing chimpanzees to support their body weight with their feet, while freeing their hands for other activities. The muscles controlling this opposable big toe are robust and powerful, contributing to overall climbing capability in ways that human feet, optimized for bipedal walking, cannot match. Bone density and internal architecture also contribute to skeletal strength, though these factors affect injury resistance more than force generation. Chimpanzees have denser bones than humans in many skeletal elements, with higher trabecular bone volume, fraction, thicker cortical bone, and more robust joint surfaces. This density helps their bones resist the large forces generated during climbing and explosive movements, reducing the risk of fractures and joint injuries. Studies using micro-CT scanning to examine trabecular bone architecture have revealed that chimpanzees show higher bone volume throughout their skeletons compared to humans. This is partly a systemic difference related to activity levels and mechanical loading, but it also reflects the higher peak forces that chimpanzee bones must routinely withstand. The bone density difference is particularly notable in the upper limbs, where chimpanzees support their body weight during climbing, and in joint surfaces like the humeral and femoral heads, where large compressive forces are transmitted between bones. The distribution of muscle mass on the skeleton also affects strength in ways that go beyond simple leverage calculations. Chimpanzees have more muscle mass in their upper limbs and shoulders compared to humans. Studies of body composition have shown that approximately 16% of a chimpanzee's total body mass is in the upper extremities, compared to only about 9% in humans. This means that not only do chimpanzees have better leverage for upper body strength, they also have more absolute muscle mass dedicated to these activities. The larger upper limb muscle mass in chimpanzees reflects their arboreal lifestyle and the demands of climbing. Supporting and moving body weight through tree canopies requires powerful arm and shoulder muscles, and natural selection has favoured individuals with greater upper body musculature. Humans, having adapted to bipedalism, shifted muscle mass to the lower limbs, with our leg muscles becoming larger and more powerful, while our arms became relatively gracile. This redistribution is appropriate for our locomotor mode, but means we have less muscle mass available for pulling and climbing activities. The relationship between muscle mass and skeletal architecture creates what biomechanists call the musculoskeletal system, an integrated entity where muscles, bones, and joints work together to produce movement. Optimizing this system requires coordinating multiple factors: muscle size and composition, moment arms and leverage, bone strength and geometry, joint ranges of motion. Evolution has tuned these factors together in both humans and chimpanzees, creating integrated musculoskeletal systems adapted for different behavioral repertoires. When we consider biomechanics, we also need to think about how forces are generated during actual movements, not just static force production. Dynamic movements involve accelerations, momentum, and energy transfer through the body. The skeletal architecture affects how efficiently these dynamic processes occur, how much of the muscular work goes into useful movement versus being absorbed in the body or dissipated in non-productive directions. Chimpanzee skeletal architecture appears well suited for dynamic pulling and climbing movements. The combination of long arms, superiorly oriented scapulae, robust muscle attachments, and appropriate moment arms creates a kinematic chain that efficiently transfers energy from muscles to hands and ultimately to the external environment. When a chimpanzee pulls on a branch to climb, the force generated by shoulder and back muscles propagates efficiently through the rigid skeletal framework to the hands, minimizing energy loss to body segment rotation or elastic deformation. Humans, with our different skeletal architecture, are less efficient at these pulling movements. Our laterally oriented scapulae, shorter arms, and different muscle attachment patterns mean that more of our muscular effort goes into stabilizing joints and less into productive pulling force. We can perform these activities. Obviously, humans can climb and pull, but we do so less efficiently than chimpanzees, requiring more muscular effort to achieve the same external work. On the other hand, human skeletal architecture excels at activities chimpanzees perform poorly. Our elongated legs and specialized foot structure make us exceptional long-distance walkers and runners. Our mobile shoulders and long moment arms for deltoid muscles make us superior throwers. Our precision grip capabilities, enabled by thumb anatomy and muscle arrangements, allow for tool use and manipulation that chimpanzees cannot match. The skeletal differences between humans and chimpanzees represent different optimization strategies, each species specializing for their particular ecological niche. The biomechanical story also connects to energy efficiency, a theme we've touched on previously. More efficient lever systems require less muscular force to achieve the same external work, conserving energy. For activities that must be sustained over long periods, this efficiency matters enormously. Humans, with our walking-optimized lower limbs, can cover vast distances with relatively little energy expenditure per kilometre. Chimpanzees, with their climbing-optimized upper limbs, can ascend trees efficiently despite working against gravity. But these specializations come with trade-offs. Human lower limb architecture that excels at walking is poorly suited for climbing. Chimpanzee upper limb architecture that excels at climbing is poorly suited for precision manipulation. Neither species can do everything well. Evolution has made choices, optimizing for some capabilities while accepting limitations in others. Understanding these trade-offs helps explain not just what each species can do, but why they behave as they do, the ecological and behavioral strategies that their anatomy enables and constrains. Recent advances in musculoskeletal modelling have allowed researchers to quantify these biomechanical effects with increasing precision. Computer models incorporating detailed skeletal anatomy, muscle properties, and movement patterns can simulate how forces propagate through the musculoskeletal system during various activities. These models can predict joint torques, muscle forces, and energy expenditures, providing insights into how different anatomical configurations affect performance. Studies using these modelling approaches have compared chimpanzee and human musculoskeletal function during tasks like climbing, pulling, and walking. The results consistently show that chimpanzees achieve better performance in upper body strength tasks not solely due to muscle properties, but through a combination of muscle characteristics and skeletal architecture working together. The model suggests that skeletal factors might contribute an additional 10 to 20% to the overall strength advantage, beyond what muscle composition and motor control provide. The modelling work also reveals which specific anatomical features contribute most to the strength differential. Moment arm lengths for key muscles like the latissimus dorsi and biceps emerge as particularly important factors. Scapular orientation matters significantly for overhead pulling activities. Arm length ratios affect both the magnitude and direction of forces during climbing movements. By isolating these factors in models, researchers can understand their individual and combined effects more clearly than possible through anatomical studies alone. One particularly interesting finding from biomechanical modelling concerns the role of body proportions in strength. Chimpanzees have relatively short legs and long arms, with an intermembral index, the ratio of arm length to leg length that differs significantly from humans. These proportional differences affect the animal's centre of mass location, and thus the moments and torques required to maintain posture and generate force during various activities. The lower centre of mass in chimpanzees, positioned more anteriorly due to their body proportions, provides inherent stability during quadrupedal stance and during pulling activities. When a chimpanzee pulls on something, their body configuration naturally resists being pulled off balance, allowing them to commit more fully to force generation. Humans, with our high centre of mass perched atop long legs, must carefully manage pulling forces to avoid losing balance, effectively constraining how much force we can safely generate in many contexts. This stability advantage compounds the strength advantages we've discussed previously. Not only do chimpanzees have stronger muscles and better leverage, they can also use those muscles and that leverage more effectively, because their body configuration allows for stable force production in a wider variety of positions and orientations. It's another example of how multiple factors interact synergistically, creating combined effects larger than the sum of individual components. The biomechanical perspective also helps explain some specific strength feats that chimpanzees are known for. For instance, chimpanzees are exceptional at vertical pulling, able to generate forces several times their body weight when pulling downward on a branch or rope. This capability stems partly from muscle strength, but the skeletal architecture contributes significantly. The superior scapular orientation, long moment arms for shoulder extensors, and stable body configuration all enhance vertical pulling capability. Humans, attempting the same task, must work against skeletal architecture optimized for different activities, resulting in lower peak forces even with intensive training. Similarly, chimpanzees excel at climbing tasks that require pulling the body weight upward against gravity. The combination of strong biceps and brachialis muscles, long moment arms for these muscles, robust finger flexors with appropriate leverage, and body proportions that keep the center of mass close to handholds all contribute to climbing efficiency. Humans can climb, obviously rock climbers and mountaineers demonstrate impressive capabilities, but we do so working against anatomy that makes climbing more difficult than it needs to be from a biomechanical perspective. The skeletal architecture story also connects to injury patterns and musculoskeletal pathology. Humans, with our unusual anatomy and unusual activity patterns, suffer from specific injuries related to our skeletal configuration. Rotator cuff tears, for instance, are common human injuries related to our laterally projecting acromion and the mechanical stresses this creates on shoulder tendons. Chimpanzees, with their different shoulder architecture, experience different injury patterns related to their different mechanical loading regimes. Understanding these species' differences in musculoskeletal biomechanics has practical applications beyond satisfying scientific curiosity. Engineers designing prosthetics, exoskeletons, and assistive devices can learn from both human and chimpanzee anatomy, understanding how different skeletal configurations optimize for different activities. Sports scientists studying human performance can identify biomechanical limitations and potentially develop training methods that work within these constraints. Evolutionary biologists reconstructing the locomotion and behavior of extinct hominins can use biomechanical principles to interpret fossil anatomy and infer behavior from skeletal remains. The fossil record of human evolution shows a gradual transition in skeletal architecture from more chimpanzee-like proportions toward more human-like proportions. Early hominins like Australopithecus had relatively long arms and short legs, similar to chimpanzees, though with modifications for bipedalism in the pelvis and lower limbs. Later, hominins like Homo erectus showed progressively more human-like proportions, with longer legs and relatively shorter arms. This anatomical transition presumably accompanied behavioral changes, with our ancestors becoming more committed to terrestrial bipedalism and less dependent on climbing. Each step in this anatomical transition involved trade-offs. As arms shortened and shoulder architecture changed, early hominins would have gradually lost some climbing efficiency and pulling strength, but they gained other capabilities: more efficient walking, longer ranges of daily travel, freed hands for carrying and tool use. The skeletal changes were part of a broader package of adaptations that collectively redefined what it meant to be a hominin versus an ape. The study of fossil hominins reveals fascinating intermediate forms that combined features of both humans and apes. Australopithecus afarensis, the species to which the famous Lucy skeleton belongs, had arm proportions intermediate between chimpanzees and modern humans. Their arms were shorter than chimpanzees but longer than modern humans, suggesting they retained some climbing ability, while also being capable of efficient bipedal walking. The skeletal architecture of these early hominins probably conferred strength capabilities intermediate between modern chimpanzees and modern humans, reflecting their mixed locomotor repertoire. Homo erectus, which appeared about 2 million years ago, shows skeletal proportions much closer to modern humans. Their arm-to-leg ratio approaches modern human values, suggesting they had largely abandoned climbing as a regular activity and were fully committed to terrestrial bipedalism. This anatomical transformation probably meant they had strength capabilities more similar to modern humans than to chimpanzees, though their overall robustness suggests they may have been somewhat stronger than modern humans, reflecting the physically demanding lifestyles of our ancestors. The evolutionary trajectory of shoulder architecture is particularly informative. Early hominins show shoulder morphology intermediate between apes and humans, with scapular orientation that was neither as superiorly oriented as chimpanzees nor as laterally oriented as modern humans. This intermediate configuration probably allowed for both overhead reaching, useful for gathering fruit from trees, and manipulation in front of the body, useful for tool use and carrying. Over time, as our ancestors became less dependent on climbing and more dependent on tool use and throwing, selection favoured increasingly lateral scapular orientation, culminating in the modern human shoulder. The archaeological record of tool use provides independent evidence for this interpretation. The earliest stone tools, dating to about 3.3 million years ago, are simple flakes and cores that could have been made with relatively ape-like hand and shoulder anatomy. But by 2 million years ago, tools become more sophisticated, showing signs of careful planning and execution that probably required more human-like manual dexterity and shoulder function. The correlation between anatomical changes visible in fossils and behavioural changes evident in archaeological remains suggests these evolved together, with anatomy enabling behaviour and behavioural advantages driving anatomical evolution. The biomechanical analysis of extinct species also raises intriguing questions about Neanderthals, our closest extinct relatives. Neanderthal skeletons show several features suggesting they were extremely robust and probably quite strong. They had thick bones, large muscle attachment sites, and skeletal proportions that differ subtly from modern humans in ways that might have conferred biomechanical advantages for strength. Some researchers have suggested Neanderthals might have been intermediate between modern humans and earlier hominins in strength. Perhaps not as strong as chimpanzees, but stronger than modern humans. Testing this hypothesis requires detailed biomechanical modelling and careful analysis of Neanderthal skeletal remains. Modern humans retain some vestiges of our arboreal ancestry in our skeletal structure. We have relatively mobile shoulder joints compared to most terrestrial mammals, a legacy of our brachiating ancestors. Our hands retain gripping capabilities, though modified for precision rather than pure strength. Our foot structure, while dramatically modified for bipedalism, still shows signs of its grasping origins in the complex arrangement of foot bones and muscles. These vestiges remind us that human anatomy is a product of history, shaped by our evolutionary journey from arboreal apes to terrestrial bipeds to tool-using, culture-bearing modern humans. The comparative study of skeletal biomechanics also reveals interesting patterns across primates more broadly. All apes, including gibbons, orangutans, gorillas, chimpanzees, and humans, share certain skeletal features that distinguish them from monkeys and other primates. These shared features, like mobile shoulder joints and reduced tail structures, reflect our common ancestry and the arboreal lifestyle of early apes. But within apes, there's tremendous diversity in skeletal architecture reflecting different locomotor strategies and ecological niches. Gibbons, the smallest apes, are specialised brachiators, swinging through trees with remarkable speed and agility. Their skeletal architecture emphasises shoulder mobility and arm length, with extremely long arms relative to body size and highly mobile shoulder joints. Their strength capabilities are impressive for their size, particularly in overhead pulling and swinging movements. Gibbons represent one extreme of ape skeletal diversity, optimised for rapid arboreal locomotion through brachiation. Orangutans, the largest arboreal apes, face different mechanical challenges than gibbons. Their large body size makes rapid brachiation difficult, so they move through trees more cautiously, using all four limbs to distribute their weight across multiple supports. Their skeletal architecture reflects this strategy, with robust bones, powerful muscles and skeletal proportions that facilitate careful climbing rather than rapid swinging. Despite their cautious movement style, orangutans are extremely strong, capable of generating enormous forces when pulling or manipulating their environment. Gorillas, the largest primates, are primarily terrestrial but retain climbing ability, particularly as juveniles. Their skeletal architecture represents a compromise between terrestrial quadrupedalism and arboreal capability. Adult male gorillas, weighing up to 200 kilograms, are too large for regular climbing, but their skeletal architecture still shows adaptations for generating enormous forces, particularly in their massive forearms and powerful shoulder muscles. Gorillas are probably the strongest primates in absolute terms, though chimpanzees may be stronger relative to body size. Understanding this diversity of ape skeletal architectures helps contextualize human evolution. We didn't evolve from chimpanzees or any other living ape. Rather, we share common ancestors with each ape lineage, and each lineage, including the human lineage, has been evolving independently for millions of years. Chimpanzees aren't frozen representations of our ancestors. They're fully modern species, as evolved and specialised as we are, just specialised for different challenges. Their skeletal architecture is optimised for their arboreal lifestyle, just as ours is optimised for our terrestrial bipedal lifestyle. Before we move to the final major factor in chimpanzee strength, let's summarise what we've learned about skeletal architecture and biomechanics. Chimpanzees have longer moment arms for many upper limb muscles, providing better leverage for pulling and climbing activities. Their skeletal architecture, including superiorly oriented scapulae, different arm proportions, and robust muscle attachment sites, optimizes force transmission during overhead activities. Their lower center of mass provides stability during force generation. These skeletal factors work synergistically with muscle composition and motor control to amplify strength, adding perhaps 10 to 20% to the overall advantage beyond what muscles and nerves provide alone. This is the third piece of the puzzle, the mechanical framework that determines how muscular force translates into actual movement and work output. But there's one more major factor we need to examine, one that ties together metabolism, body composition, and overall muscular capacity. We need to understand how chimpanzees allocate their body mass between muscle, fat, and other tissues, and how this allocation affects their strength capabilities. Let's examine the fourth and final major factor in chimpanzee strength, their unique body composition. Part 4. Body composition and muscle mass distribution. If you've ever seen an image of a hairless chimpanzee, perhaps one suffering from alopecia, or prepared for anatomical study, you've witnessed something striking. The animal appears to be made entirely of muscle. Every contour of the body reveals muscular definition. Shoulders bulge with deltoids and trapezius. Arms show clearly delineated biceps, triceps, and forearm muscles. The chest displays pectoralis muscles that would make a bodybuilder envious. The abdomen is a washboard of abdominal muscles with essentially no fat covering. These animals look like they've been training at the gym for years, achieving a level of muscularity that humans can only approximate with dedicated exercise and strict dietary control. But here's the remarkable thing. Wild chimpanzees don't train at gyms. They don't follow carefully calibrated exercise programs or monitor their body composition. They don't consume protein supplements or count macronutrients. They simply exist, foraging, traveling, socializing, and occasionally engaging in bursts of intense activity. And yet their bodies maintain this extraordinary level of muscularity as a baseline, as their normal condition. This brings us to the fourth and final major factor in understanding chimpanzee strength: their body composition, the ratios of muscle, fat, and other tissues that make up their total body mass. Let's start with the most striking aspect of chimpanzee body composition, their near absence of body fat. When researchers at Stony Brook University conducted comprehensive anatomical dissections of 13 bonobos, one of the 2 chimpanzee species, they measured body composition with unprecedented precision. The results were startling. Female bonobos had an average body fat percentage of approximately 3.6%. Male bonobos averaged just 0.05%, essentially zero measurable fat. To put these numbers in perspective, consider that healthy adult human males typically maintain body fat percentages between 12 and 20%. Human females maintain between 20 and 30%. Even elite male athletes, bodybuilders preparing for competition, or endurance runners at peak fitness rarely drop below 5% body fat. And when they do, it's considered potentially unhealthy, a temporary state achieved through extreme measures that cannot be sustained long-term without negative health consequences. Chimpanzees maintain these extraordinarily low body fat levels as their normal healthy baseline. Male chimpanzees in the study, regardless of age, body mass, or whether they lived in captivity or the wild, all measured at essentially zero body fat. The negligible fat they did possess was located primarily in specific anatomical sites like the cheeks, groin, and around certain organs, serving essential physiological functions that cannot be eliminated. Apart from these minimal necessary deposits, male chimpanzees are virtually fat-free. Female chimpanzees show slightly higher body fat levels, but still remarkably low by human standards. The 3.6% average comes primarily from reproductive females, those who were pregnant or lactating at the time of the study. These females had body fat percentages as high as 8.6%, extra energy stores needed to support the metabolic demands of reproduction and nursing. Non-reproductive females had body fat levels comparable to males, effectively zero. This near absence of body fat has profound implications for strength, both directly and indirectly. Directly, it means that a much higher percentage of a chimpanzee's body mass is muscle. When you compare a 60-kilogram chimpanzee to a 60-kilogram human, the chimpanzee is carrying perhaps 50 kilograms of muscle, compared to perhaps 35 or 40 kilograms in the human, with the difference made up by the human's higher fat mass. More muscle means more strength, all else being equal. But the effect is more than just having more muscle mass. The absence of body fat also affects how muscle fibers are organised and function. Human muscles, even in relatively lean individuals, contain intramuscular fat, adipose tissue, interspersed between muscle fibers and within the connective tissue surrounding fiber bundles. This intramuscular fat can account for several % of muscle volume, space that could otherwise contain contractile tissue. Chimpanzee muscles have minimal intramuscular fat, essentially none, meaning that a chimpanzee muscle of a given size contains more actual muscle fibers than a human muscle of the same size. The metabolic implications of low body fat are also significant. Fat tissue is metabolically expensive to maintain, requiring blood flow, innervation, and cellular maintenance. By minimizing body fat, chimpanzees free up metabolic resources that can be allocated elsewhere. Some of this saved energy presumably goes into maintaining and operating their more substantial muscle mass. Some might support other physiological systems. The point is that body composition affects not just the structural properties of the body, but also its metabolic economy, influencing how energy is distributed among competing demands. The evolutionary reasons for this dramatic difference in body composition between humans and chimpanzees likely relate to diet, activity patterns, and the different selective pressures faced by each species. Chimpanzees in the wild maintain high levels of physical activity throughout their lives. They forage actively, travel through forest canopies, engage in social behaviours that often involve physical exertion, and occasionally hunt. This activity level, sustained day after day, year after year, maintains high muscle mass and prevents fat accumulation. Their diet also differs significantly from typical human diets, especially modern human diets. Chimpanzees are primarily frugivorous, consuming large quantities of fruits supplemented with leaves, insects, and occasionally meat. This diet is high in fiber and relatively low in caloric density, meaning chimpanzees must consume large volumes of food to meet their energy needs. The combination of high activity levels and a diet that doesn't readily promote fat storage makes it difficult for chimpanzees to accumulate body fat, even when food is abundant. Humans evolved under different conditions, particularly after our ancestors left the forests and adapted to more open, seasonal environments. The ability to store body fat became advantageous, providing a buffer against periods of food scarcity. Individuals who could build fat reserves during times of abundance were more likely to survive lean periods, passing their genes for efficient fat storage to subsequent generations. Over millions of years, natural selection favoured humans with greater capacity for fat storage, particularly in females where fat reserves support the enormous metabolic demands of pregnancy and lactation. Modern humans have taken this evolutionary adaptation to an extreme, with many populations now maintaining body fat levels far higher than would have been typical for our ancestors. This is partly due to dietary changes, consuming calorie-dense processed foods that didn't exist in our evolutionary past, and partly due to reduced physical activity levels compared to the constant exertion typical of hunter-gatherer lifestyles. But even traditional human societies that maintain high activity levels and consume traditional diets show body fat percentages well above chimpanzee levels. The capacity for fat storage appears to be a fundamental human characteristic, not solely a product of modern lifestyles. The difference in body composition also connects to reproduction and development. Human infants are born with substantial body fat, approximately 15% of body mass, much higher than infant chimpanzees. This fat supports the rapid brain growth that characterizes human development, providing energy reserves that can be mobilized when food intake is insufficient. Human breast milk is also much higher in fat content than chimpanzee milk, supporting continued fat deposition during nursing. These differences in infant body composition reflect the different developmental strategies of the two species, with humans investing heavily in brain development at the cost of extended vulnerability and dependence. The near absence of body fat in male chimpanzees is particularly striking and raises questions about how they fuel intense activities. Fat is a dense energy store, containing about 9 calories per gram compared to about 4 calories per gram for carbohydrates or protein. Without fat reserves to draw upon during prolonged exertion, chimpanzees must rely primarily on stored carbohydrates, glycogen in the muscles and liver, or on immediate energy intake from food. This probably constrains their activity patterns, favouring brief intense efforts that can be fueled by glycogen, rather than sustained efforts that would require mobilizing stored fat. research on wild chimpanzees has shown that they generally don't engage in prolonged endurance activities. They don't travel long distances in single bouts, instead interspersing travel with rest and foraging. They don't sustain maximum effort for extended periods, instead operating in bursts of activity followed by recovery. This activity pattern matches what we'd expect given their body composition and energy storage capacity. Without fat reserves, sustained high-intensity activity becomes metabolically challenging, potentially depleting glycogen stores and forcing the breakdown of muscle protein for energy, which would be counterproductive. Humans, with our substantial fat reserves, can sustain activity much longer. Even a lean human with 12% body fat carries about 9 kilograms of fat in a 75 kilogram body, representing roughly 80,000 calories of stored energy. This is enough to fuel days of continuous moderate activity or many hours of intense exercise. Fat oxidation provides sustained energy during prolonged efforts, allowing humans to walk all day, run for hours, or perform repetitive work without depleting glycogen and breaking down muscle tissue. The body composition story also involves the distribution of muscle mass throughout the body, beyond just total muscle quantity. We've already mentioned that chimpanzees carry a higher percentage of their body mass in their upper extremities, about 16% compared to 9% in humans. This upper body muscle distribution reflects the different demands placed on arms and shoulders in the two species. Chimpanzees use their arms for locomotion, climbing and occasionally brachiating through trees, as well as for manipulation and fighting. Supporting body weight and moving through three-dimensional arboreal environments requires powerful arm and shoulder muscles. Individual muscle sizes within the upper limb also differ between chimpanzees and humans. Anatomical studies have shown that despite chimpanzees' smaller average body size, many of their upper limb muscles are absolutely larger than the corresponding human muscles. The latissimus dorsi, the large back muscle that pulls the arm downward and backward, is substantially larger in chimpanzees. The pectoralis major, the chest muscle involved in arm movements, is also relatively larger. The biceps, triceps, and many forearm muscles show similar patterns, with chimpanzee muscles equaling or exceeding human muscle sizes despite the overall smaller body mass. This means that when comparing similarly sized individuals, say a 60 kilogram human and a 60 kilogram chimpanzee, the chimpanzee has considerably more muscle mass in the arms and shoulders. They're starting with a larger engine, so to speak, more contractile tissue available to generate force in pulling and climbing activities. When combined with the favourable muscle composition, motor control, and skeletal leverage we've already discussed, these larger upper limb muscles contribute substantially to overall strength. The lower limb muscle distribution shows the opposite pattern. Humans carry more muscle mass in our legs than chimpanzees do, reflecting our adaptation to bipedal locomotion. Our quadriceps, hamstrings, gluteal muscles, and calf muscles are all relatively larger than the corresponding chimpanzee muscles. This makes sense given that human legs must support and propel body weight throughout daily bipedal activities, while chimpanzee legs serve primarily for quadrupedal locomotion and climbing, with less demand for power generation. Studies of body composition using techniques like dual-energy X-ray absorptiometry have quantified these muscle distribution differences precisely. When researchers scanned human and chimpanzee bodies, measuring muscle and fat distribution across body segments, the results confirmed the upper versus lower body pattern. Chimpanzees showed higher muscle mass in arms, shoulders, and upper trunk, while humans showed higher muscle mass in hips, thighs, and calves. The total muscle mass might be similar between species of equal body weight, but the distribution differs markedly, reflecting different functional demands. This redistribution of muscle mass represents an evolutionary trade-off. As our ancestors became committed to bipedalism, natural selection favoured individuals with powerful legs capable of efficient walking and running. This came at the cost of arm and shoulder strength, with muscle mass shifting from the upper body to the lower body. The transformation probably occurred gradually over millions of years, tracking the anatomical changes we can observe in the hominin fossil record, with progressively more human-like body proportions appearing as bipedalism became more central to hominin ecology. The muscle distribution differences also affect centre of mass location and movement mechanics. With more muscle mass in the upper body, chimpanzees have their centre of mass positioned more anteriorly and superiorly than humans. This affects balance and stability during various activities. It also affects the moments and forces required for different movements, influencing which activities are energetically favourable and which are costly. For instance, chimpanzees' forward centre of mass makes quadrupedal locomotion relatively efficient but bipedal walking more difficult. They must work constantly to maintain upright posture against a centre of mass that wants to pull them forward. Humans, with our centre of mass positioned more posteriorly and inferiorly due to our heavier legs, find bipedal walking natural and energetically efficient, but would struggle with extended quadrupedal locomotion. The differences in body composition between chimpanzees and humans also have implications for thermoregulation and climate adaptation. Body fat serves as insulation, helping maintain core body temperature in cold environments. Humans, with our substantial subcutaneous fat layer, can maintain body temperature across a wide range of environmental conditions. This insulation capacity probably contributed to human success in colonizing diverse environments, including temperate and Arctic regions where chimpanzees could never survive. The ability to store fat and use it for both energy reserves and thermal insulation gave humans flexibility and habitat selection that other primates lack. Chimpanzees, with their minimal body fat, have less thermal insulation and are more vulnerable to cold stress. They're restricted to tropical and subtropical environments where temperatures rarely challenge their thermoregulation. When temperatures drop, chimpanzees must increase metabolic heat production or seek behavioural solutions like huddling together or finding sheltered locations. They lack the physiological buffer that human body fat provides, making them more constrained in habitat selection. The loss of body hair in humans also connects to body composition and thermoregulation. Humans have far less body hair than chimpanzees, essentially naked compared to our fur-covered relatives. This hair loss probably occurred as an adaptation for evaporative cooling during sustained exertion in hot environments. With reduced hair coverage, humans can dissipate heat more effectively through sweating, allowing for extended physical activity that would cause chimpanzees to overheat. The combination of reduced hair and increased body fat represents a different thermoregulatory strategy, one adapted for different thermal challenges than chimpanzees face. The body composition differences also influence dietary requirements and feeding behaviour. Maintaining large amounts of muscle requires substantial protein intake, because muscle tissue undergoes constant breakdown and rebuilding. Chimpanzees, with their high muscle mass and low body fat, need regular protein consumption to maintain their musculature. This partly explains why chimpanzees supplement their primarily frugivorous diet with insects, which are rich in protein and occasionally hunt other animals. The protein from these sources supports muscle maintenance and growth. Humans can maintain muscle mass on more varied diets, including largely plant-based diets, partly because our lower muscle mass requires less absolute protein. Additionally, the shift towards slow-twitch muscle fibers in humans means our muscles undergo less damage during use compared to the fast-twitch dominant muscles of chimpanzees. Slow-twitch fibers are more resistant to contraction-induced damage, requiring less frequent repair and thus less dietary protein. This dietary flexibility allowed human ancestors to thrive in diverse environments with different food availability. The body composition story also connects to aging and life history. Humans show characteristic changes in body composition as we age, generally losing muscle mass and gaining fat mass in processes called sarcopenia and age-related obesity. These changes are considered pathological in modern contexts but may have had adaptive significance in our evolutionary past, allowing older individuals to maintain energy reserves despite reduced physical capabilities. The increased body fat in older humans provides an energy buffer that could have been important for survival in our ancestors. Chimpanzees show different aging patterns. While they do lose some muscle mass with age, they maintain remarkably lean body composition throughout their lives. Older chimpanzees remain muscular and relatively strong, often continuing to hold positions of dominance in their social groups well into middle age. The preservation of muscle mass and strength into later life probably reflects the ongoing importance of physical capability in chimpanzee social dynamics, where dominance hierarchies are often maintained through displays of strength and occasional physical confrontations. The captive versus wild difference in chimpanzee body composition is also worth noting. While muscle fiber type distributions appear stable between wild and captive populations, body composition can change with captivity. Captive chimpanzees, provided with abundant calorie-dense food and reduced opportunities for natural activity patterns, can develop higher body fat percentages than their wild counterparts. Some captive chimpanzees have been observed with body fat percentages approaching 10 or even 15%, still low by human standards, but notably higher than wild chimpanzees. This suggests that while chimpanzees have a genetic baseline favouring minimal body fat, their body composition is somewhat plastic, responsive to diet and activity levels. Even so, captive chimpanzees rarely approach human levels of body fat, even when provided with unlimited calorie-dense foods and minimal forced exercise. They appear to have metabolic set points that resist substantial fat accumulation, perhaps through increased thermogenesis, higher baseline metabolic rates, or spontaneous activity that burns excess calories. The research into chimpanzee body composition has also revealed interesting details about organ sizes and their contribution to metabolic demands. Human brains, as we've mentioned, are approximately 3 times larger than chimpanzee brains in absolute size, despite similar body masses. This brain size difference represents a substantial allocation of body mass, with humans devoting a larger percentage to neural tissue. The metabolic cost of supporting our enlarged brain is enormous, consuming roughly 20 to 25% of our resting metabolic rate. Chimpanzees, with their smaller brains, devote less of their metabolic budget to neural tissue and more to other systems, including their muscular system. The energy saved by having a smaller brain can be allocated to maintaining and operating more muscle mass. This represents another aspect of the evolutionary trade-off between cognitive capability and physical strength. Humans traded muscle for brain, becoming weaker but smarter. Chimpanzees maintained more muscle at the cost of cognitive development, remaining stronger but with less sophisticated cognitive capabilities. The digestive system also differs between humans and chimpanzees in ways that affect body composition and energy availability. Chimpanzees have larger colons relative to body size, adapted for fermenting the fibrous plant material that dominates their diet. This large colon allows them to extract nutrients from food that humans would find difficult to digest. However, the energetic cost of maintaining this larger digestive system is substantial, representing another metabolic trade-off in their physiology. Humans have smaller colons but larger small intestines, reflecting our shift toward more calorie-dense, easier to digest foods. Cooking, which probably became common in human ancestors 1 to 2 million years ago, makes food easier to digest and increases nutrient availability. This dietary shift allowed humans to support larger brains and changed our digestive system anatomy in ways that further differentiate us from chimpanzees. The body composition differences we observe today reflect these deeper differences in metabolic strategy and dietary adaptation. Before we move to the conclusion, let's summarize what we've learned about body composition. Chimpanzees maintain extraordinarily low body fat percentages, essentially 0 in males and about 3 to 4% in non-reproductive females, compared to 12 to 30% in healthy humans. This means a much higher percentage of chimpanzee body mass is muscle, directly contributing to strength. Additionally, chimpanzees carry 16% of their body mass in upper extremities compared to 9% in humans, with individual upper limb muscles often larger than corresponding human muscles despite smaller overall body size. These body composition differences, combined with the muscle fiber, motor control and skeletal factors we've discussed, create the full picture of why chimpanzees are so remarkably strong. Conclusion. We've now explored all 4 major factors that contribute to chimpanzee strength: muscle fiber composition, motor control architecture, skeletal biomechanics, and body composition. Each factor independently contributes to their superior strength, but more importantly, these factors work together synergistically, amplifying each other's effects to produce the remarkable capabilities we observe. Start with muscle fiber composition. 67% fast twitch fibers in chimpanzees compared to 30% in humans. This alone accounts for much of the strength differential, providing muscles that can generate approximately 1.35 times more dynamic force and power. Layer on motor control differences, larger motor units, potentially less neural inhibition, more automated control patterns. These neural factors ensure that the powerful muscles are activated, effectively recruiting large fractions of muscle mass quickly and forcefully. Add skeletal biomechanics, longer moment arms, superiorly oriented scapulae, body proportions that provide stability and efficient force transmission. The skeletal architecture amplifies the force generated by muscles, converting muscular effort into external work more efficiently than human anatomy allows. Finally, consider body composition. Near zero body fat, high muscle-to-body mass ratio, concentration of muscle mass in the upper limbs. These factors ensure that a chimpanzee of a given size has more actual muscle tissue generating force than a human of equivalent weight. When you multiply these effects together, you get the observed strength differential. A chimpanzee generates 1.35 times more force from their muscle composition, perhaps another 10% from motor control advantages, another 10 to 20% from biomechanical leverage, and has more absolute muscle mass from their body composition. The combined effect produces animals that can consistently generate 1.5 times the force of humans in pulling and jumping tasks, sometimes approaching twice human strength in specific contexts like vertical pulling where all their advantages align. The 1.5 times multiplier that emerges from careful scientific studies is less dramatic than the old stories of chimpanzees being 5 or 8 times stronger than humans, but it's still remarkable. It means that an average chimpanzee can match or exceed the strength of much larger, stronger humans. It means that even trained human athletes cannot compete with chimpanzees in activities that favour explosive power. But here's the crucial insight that emerges from this investigation. Chimpanzees aren't superior to humans. They're differently adapted. Every advantage they possess in strength came at a cost, a trade-off that limited other capabilities. Their fast-twitch dominant muscles make them weaker in endurance activities. Their larger motor units reduce fine motor control. Their skeletal architecture optimized for climbing compromises efficiency in other movements. Their near-zero body fat limits sustained activity and energy buffering. These aren't flaws. They're the inevitable consequences of evolutionary specialization. Humans made different trade-offs. We gave up explosive strength for endurance. We sacrificed all-or-nothing power for fine motor control. We exchanged climbing efficiency for walking efficiency and throwing capability. We developed body fat stores and reduced muscle mass to support larger brains and buffer against food scarcity. Each trade-off made us weaker in the specific ways chimpanzees are strong, but enabled capabilities that proved even more valuable for survival in our ecological niche. The human story is one of cognitive solutions compensating for physical limitations. We're weaker than chimpanzees, but we developed weapons that made our weakness irrelevant. We lack explosive power, but we developed tools that amplified our strength. We can't climb as efficiently, but we learned to build shelters and structures. We tire more quickly in intense bursts, but we can travel farther and work longer. Our physical limitations drove innovation, forcing us to solve problems through intelligence rather than brute force. Understanding chimpanzee strength also provides a humbling perspective on human capabilities and human evolution. We tend to think of ourselves as the pinnacle of evolution, superior in every meaningful way. But the comparison with chimpanzees reveals that we're not superior across all dimensions. We're specialized, adapted for specific capabilities, impressive in some domains but limited in others. Evolution doesn't produce better organisms. It produces organisms better adapted to their specific environments and ecological niches. The story of chimpanzee strength is ultimately a story about biological diversity, evolutionary trade-offs, and the complex relationship between form and function. It reveals how subtle differences in cellular composition, neural architecture, skeletal geometry, and body composition can produce dramatic differences in capability. It demonstrates how evolution optimizes organisms for specific challenges through integrated changes across multiple biological systems. As we conclude this exploration, we're left with a deeper appreciation for both chimpanzees and humans. Chimpanzees for their remarkable physical capabilities, the product of millions of years of natural selection optimizing their bodies for arboreal life, climbing, and explosive power. Humans for our unique evolutionary trajectory, the path that led us to sacrifice strength for endurance, precision, and ultimately cognitive capability. Neither species is superior. Both are successful, adapted to their niches, remarkable products of evolutionary processes. Thank you for joining me on this exploration. I hope you found it as fascinating as I have. If you enjoyed this investigation into the biology of chimpanzee strength, please consider liking this video and subscribing to the channel. Feel free to leave comments sharing your thoughts or suggestions for future topics. Until next time, Stay curious, keep questioning, and never stop exploring the remarkable world around us. There's always more to learn, always deeper understanding to pursue, always new perspectives to gain. The journey of understanding never really ends. Good night, everyone.